All right, so the membranes on the plasma membranes of the axon terminals of the neurons in the skeletal muscle fibers are said to be excitable, and that just means that they can have depolarization and repolarization. And these events create action potentials, and action potentials are essentially electrical impulses. So why do the skeletal muscle fibers contract? And it's because the motor neurons are essentially being stimulated. And they are the stimulation of those neurons is stimulating the muscle fibers. Okay, so what is a neuromuscular junction? Just think about the words in it. Okay, it's a synapse, the junction part's a synapse between a neuron and a skeletal muscle fiber. Okay, so that axon terminal and that motor neuron is going to release neurotransmitter. And that's going to go into the synaptic cleft. So that neurotransmitter is called acetylcholine, or ACH. And so what does ACH do? Well, it binds to and opens up a chemically gated sodium channel on the muscle fiber. And sodium is going to enter the cell at that point because those sodium channels are now open. And it depolarizes what we call the motor end plate, which is basically that plasma membrane on the skeletal muscle fiber. And that generates an action potential. Okay, and we'll see what that looks like. All right, so there's your neuromuscular junction. So here's the axon terminal of the neuron, and then here's the motor end plate of that skeletal muscle fiber. The space between the two is called a synaptic cleft. Okay. So the cytoplasm in this axon terminal has vesicles in it. Okay, remember vesicles from chapter 3? So these vesicles have acetylcholine inside of them. And again, acetylcholine is a neurotransmitter, okay? Changes the permeability of the skeletal muscle fibers plasma membrane. So you also have some molecules of acetylcholinesterase here that help to break down acetylcholine, okay? So you have some homeostasis built in there. All right, so if you have a electrical impulse or action potential traveling down that axon of that neuron, and it gets to the axon terminal, okay, it's going to basically send a signal to that neuron to release acetylcholine, the neurotransmitter, from its vesicles, from a process that we learned about in chapter 3. So if you're releasing things out of the cell, the vesicles, what do we call that? You might remember? Exocytosis, right? Okay. So that action potential triggers that exocytosis, releases acetylcholine into the synaptic cleft. So those acetylcholine molecules are going to diffuse across the synaptic cleft. And then some of those acetylcholine molecules will bind to receptors on these chemically gated sodium channels, or sodium ion channels. And when that happens, essentially those sodium ion channels are unlocked, okay? And so they open up. And the concentration of sodium is going to be higher outside the cell. And so sodium is going to travel inside the skeletal muscle fiber. So, because remember diffusion, moving from high concentration to low concentration, remember that? So this is facilitated diffusion because it's using a sodium channel. So sodium ions rush in, generates an action potential in the skeletal muscle fiber, in the sarcolemma, right? The plasma membrane of this skeletal muscle fiber. Now, if those acetylcholine molecules were just there forever, those sodium channels would be open forever, right? Because they'd be attached to those receptors on those channels. So you have to have a way to be able to turn that action potential off, and that's what the acetylcholine esterase does, or ACHE. So with the ACHE, it's going to break down acetylcholine into acetic acid and choline, so it splits it apart. And that closes down those sodium channels. All right, next concept. Excitation, contraction coupling. So this is another one of those potential short answer type questions or essay questions you'd have to explain. So, and that's the term that just means you pair excitation with contraction, okay? So with action potentials, they're traveling down the T tubules. So like we just learned about action potential getting basically you get an action potential once the acetylcholine makes the sodium rush into the skeletal muscle fiber. Now, that action potential is then going to travel down the T tubules to the triads. Remember, the triads are essentially the T tubule and T terminal cisternae on either side. And when that happens, calcium ions get released from the terminal cisternae of the sarcoplasmic reticulum. And that those calcium ions are then going to bind to troponin. And troponin is going to change the shape of troponin. So the calcium ions would make troponin change shape. Now, there's something called a troponin-tropomyosin complex, and it's going to move or change position when that calcium ion binds to the troponin. And when that happens, active sites on the thin filaments, your actin, get exposed. And that initiates the contraction cycle. So first you have to have excitation, and that excitation leads to contraction, okay? So it's getting the muscle fiber ready to contract. All right. So again, skeletal muscle fiber is going to contract because that motor neuron stimulated it at the neuromuscular junction. And the stimulus is an action potential, okay, at the axon terminal. So when you get excitation, that action potential makes that acetylcholine get released into the synaptic cleft, right? We just learned about that. Causes excitation, produces an action potential in the sarcolemma, the cell membrane of the muscle fiber, which leads to releasing calcium ions from those terminal cisternae. Because remember, the action potential is going to go down along the sarcolemma and down the T tubules. And once it hits the T tubules, it hits the triads, right? So, and that touches the terminal cisternae, makes them release their calcium. And you get contraction cycle beginning. Calcium ions then, once they release from the terminal cisternae, are going to bind to troponin. Okay, exposes the active sites on your thin filaments. It allows cross bridges on the myosin to bind to the actin. And this happens as long as you have ATP. Okay, you need energy for this to happen. So your sarcomeres will shorten. Your thin and thick filaments are going to interact, pulls the end of the muscle fiber together. Okay, so your sarcomere, Z-line to Z-line, gets shorter. Then you get muscle tension, okay? Produces a pull or tension on those tendons. So something that you see at the cell level, at the muscle fiber level, is affecting the organ level, okay? Because the muscle is then pulling on the tendon. All right, so neuromuscular junction. So you get your contraction cycle. It begins, you get active site exposure. And you form cross bridges. And that happens when myosin will bind to the actin. Then the myosin head's going to pivot. We call that a power stroke. Then that cross bridge is going to detach. Okay, the head's going to detach. Then you reactivate the myosin so you can potentially do it again. So again, this is another set of things that you want to be able to explain. Okay? All right, so contraction cycle begins. There's that calcium. It was released from the terminal cisternae. Okay. This is in the zone of overlap. Okay. So remember, you have your actin and your myosin, right? Your thin and thick filaments. You have an area where those two proteins overlap each other. So your calcium ions are going to bind to troponin. And it weakens the bond between actin and this troponin-tropomyosin complex. And troponin then is going to move in position, and it rolls the tropomyosin away from the active sites on the actin. And the actin are like the red, essentially the red balls, okay? And so the tropomyosin essentially, okay, you've got the strands and the troponin are these orange balls. Okay, so when calcium binds, moving the tropomyosin out of the way. So now the heads of the myosin can attach to these areas or active sites on the actin. Then you get what's called a cross bridge, where the myosin head attaches to the active site of the actin. All right, then the myosin head is going to pivot in position. And the energy that was stored in the resting state is going to get released. So it's stored here and here, and it's getting released here and here. And by pivoting, you're pulling the sarcomere closer together. And that, we call that a power stroke when that happens. And you're releasing, you had ATP, now you're releasing ADP and phosphate. Okay, cross bridge is going to detach. Get another ATP attached to the myosin head, and that causes the detachment of the myosin from the actin. Now your active site's exposed, you can get another cross bridge. And it's still exposed because you still have calcium bound to the troponin-tropomyosin complex. Okay. All right, then you reactivate the myosin by bringing it back to its original position. Okay. All right. So I think this is a good place to stop. So first thing I'd like for you to do is discuss with your partner excitation contraction coupling. Okay, discuss that. And you can do that at home as well. And if you're watching live and you want to type that, those steps there in the chat, you can do that as well. Next thing I'd like for you to explain is the contraction cycle. Okay. All right, so let's go back. So resting the sarcomeres, so let's kind of review what we talked about last class. This is your thick filament, okay? This is myosin, this is myosin, the thick filament. The red, this is actin, okay? The green is titin, helps to stabilize actin and myosin. The middle of the thick filament is the M line. Okay, area where there are only thick filaments is called the H zone. The area where you have thick end filaments is called zone of overlap. And the zigzag line here in the thin filaments, that's your Z line. So when the sarcomere is at rest, that myosin head is said to be already energized. Okay, it has the power, it has the energy it needs to have a contraction. So each myosin head is actually pointing away from the M line. You see that? It's pointing away from the M line. And in that position, the myosin head is essentially cocked like the spring in a mousetrap. So cocking the myosin head requires some energy. And you do that by breaking down ATP. And so in a sense, you could say the myosin head functions as an ATPase, because ATPase, anytime you see ACE, usually it's something that breaks whatever that is that precedes it down. And so when the contraction cycle starts, your products, ADP and phosphate, represented as P, are still bound to the myosin head. Okay. Now in the contracted or contracted sarcomere, what's different? Okay, so you can see here, relative to this one, you have a greater zone of overlap. Does everybody see that? There's more overlap of the actin and the myosin, the thick and the thin filaments. So the entire cycle gets repeated several times a second, okay? As long as your calcium ion concentrations are high enough and you have ATP. So the calcium ion levels get elevated as long as you have action potentials passing along the T-tubules and telling them to stimulate the terminal cisternae to release their calcium. So once that stimulus gets removed, calcium ions or calcium channels in the sarcoplasmic reticulum close. Your calcium ion pumps start pulling the calcium from the cytosol, start storing it back in the terminal cisternae. So there's some homeostasis there. Your troponin molecules will then shift position and swing your tropomyosin, the strands, over the active sites. So you get no more cross-bridge formation because the active sites are closed. So you have to have your calcium, you have to have your ATP for the contraction. But also want to make sure that I indicate to you in the contraction, I don't know if you remember this from last class, but I think we said that basically the myosin are the dark bands and the thin filaments are the thin filaments of the actin. Those are your light bands. Okay, so your dark band or your thick filaments, they stay the same length. Like myosin didn't change that really changed is the ends of the sarcomere got pulled closer together, right? So the distance from Z line to Z line shortened. Your zone of overlap increased. Essentially, your H zone decreased because of the increased overlap. Okay, but the dark bands actually are still the same length. Okay. So your thick filaments stay the same size. This is what I want to make sure you understand. All right, so in this whole process of contraction, how do we get muscle tension? So when you have contraction, you have pulling, okay, and that creates tension. So in order for you to move your muscles, you had to overcome the resistance, okay, or what we call the load. So, if you think about, a lot of times people do weight training and they call that resistance training, right? Because you're pushing against something that's resisting your motion. So the entire muscle is going to shorten at the same speed or same rate. Why? Because all the sarcomeres are shortening together. And how fast or how low that shortening takes place depends on what we call the cycling rate. And that just means how many power strokes did you get per second. So how many of those times of those myosin heads getting moved? So duration of your contraction is going to depend on a few things. Well, how long is your stimulus? Okay, if you have a longer stimulus, you'd have a longer contraction theoretically. Do you have enough calcium in the cytosol? Okay, is there available calcium to bind to that tropin and tropomyosin complex? Do you have ATP? If you run out of energy, you're obviously not going to be able to sustain a contraction, right? Your muscle's going to get tired. Okay, so as calcium then gets pumped back into your sarcoplasmic reticulum, and your calcium ion concentration in your cytosol goes down, your calcium ions are then going to detach from troponin. And your troponin is going to return back to its position. Then the active sites on the actin get covered again by tropomyosin. And then your contraction is over. Okay, so there's some, again, homeostasis. Because it's theoretically you really don't want for that contraction to go on forever, right? Because then that's like a bad cramp, right? But if you're able to get rid of your calcium, then you're able to end the contraction. Okay. So what happens when that doesn't work properly? So rigor mortis is one example. Okay, so your body ends up in a fixed muscular contraction after the person's died. And this is going to happen when you run out of ATP and the ion pumps stop functioning, but you get calcium ions built up in the cytosol. Okay, so there's calcium present and no ATP. So because those calcium ions are present, right, it's going to bind to the troponin tropomyosin complex, open up those active sites. Myosin is going to bind to actin, and essentially you're stuck in a place of contraction. All right, so this is steps that initiate the contraction. Kind of review here. Again, release that acetylcholine. Bind to those ACH receptors in the motor end plate. Action potential releases or reaches the T-tubule, signals the sarcoplasmic reticulum, the terminal cisternae to release their calcium. That calcium exposes active sites on the actin. You form a cross bridge of myosin head attaching to actin, and your contraction cycle is going to start. So how do you end the contraction? Well, you can break down your acetylcholine. Sarcoplasmic reticulum can reabsorb the calcium, cover up the active sites, then end the cross bridges. Okay, no more cross bridge, no more contraction. You get muscle relaxation, and that sarcomere lengthens. Okay, you end up with less overlap. All right, so let's go ahead and describe to your partner the steps that begin the muscle contraction. Steps that begin the muscle contraction. All right, not hearing enough conversation. I hear some pockets where I hear conversation, but there's some areas where I hear none. So let's make sure, even if you don't know everything, let's just say what you do know. So don't be shy. It's okay to explain something to the person sitting across from you. They probably don't have all the answers either. It's okay. Okay, so let's talk about the steps in ending the muscle contraction.
That's what I was saying.
But I think they closed it. Okay. So let's talk about, yeah.
So is it the excitation contraction, then the steps that initiate a muscle contraction, and then the contraction?
Basically, yes. So they kind of combine like in the steps to initiate. So basically there's excitation, contraction, relaxation. But, you know, and then also keeping in mind that in the contraction, it's a cycle of continued contraction. And then you get relaxation.
So there is or isn't like a specific at all?
Well, I mean, there's a specific order. I would say you have excitation. So you're having that, then you have contraction cycle, and then you have relaxation. Okay. So let's go talk a little more about tension. So here are the number of contracting sarcomeres. So how many sarcomeres you have contracting in that muscle fiber is actually a fixed number. So essentially a skeletal muscle fiber is either creating tension or it's not. Okay. So how much tension you produce? Well, the muscle's tension is going to depend on how many power strokes, right? So how many times that myosin head moves, the length of that fiber at the time it was stimulating. Okay, so because if it was completely relaxed, it has a lot of room to move. But if it was only partially relaxed, it's not necessarily going to move as far. How fast or what's the frequency that that fiber is being stimulated? You get stimulation not very often, you get not much tension, but if you get a lot of stimulation, you get more tension. So there's actually a relationship between length and tension. So how much tension you get in that muscle fibers is related to the length of the sarcomere. And kind of like what we just talked about, amount of tension is related on those number of power strokes performed. And the amount of overlap, okay, between the thin and thick filaments. So if you have potential for creating more overlap, so if the zone of overlap is short to begin with, you have potential for more overlap, for more tension. So the maximum amount of tension that you make gets produced when you get the most cross bridges, right? So you get more cross bridges, you get more tension. So when you have more cross bridges, you actually increase the zone of overlap, right? Is that those cross bridges, those power strokes are moving the actin closer to the M line. All right. So here you have a short resting length, okay? Because the sarcomere is already very contracted. The thin filaments are already extending across the center of the sarcomere, and they interfere with basically the normal orientation of the filaments. So you get low tension production. And when your thick filaments are in contact with the Z lines, so here the thick filaments, if they're touching the Z lines, you can't shorten the sarcomere at all. And the myosin heads can't pivot, and you can't produce any tension. All right, so you get the most amount of tension when your zone of overlap is very large, okay? But the thin filaments do not extend across the center of the sarcomere. Okay? But if your sarcomeres get stretched too far, you're reducing your zone of overlap too much. And so you don't have very many opportunities for cross bridges. Because here, essentially all of the myosin heads have an opportunity to have an interaction. So when the zone of overlap gets reduced to zero, so there's no connection there between actin and myosin. The thin thick filaments can't interact at all. So you get no tension. All right, this is normally prevented by the titin, which is connected to the Z lines. It connects the thick filaments to the Z lines. But if something damages your titin, the actin and myosin can get too far apart. So you want some cross bridging. You don't want too much, and you don't want not enough. It has to be just right. Okay. All right, so frequency of the stimulation. And we'll learn about some of this in lab too, so you'll get to see this as well there when we talk about muscular system. But single neural stimulation is going to create what we call a twitch, and that's a single contraction between 7 and 100 milliseconds. Okay, you're going to get sustained muscle contraction. That in order to have that, you have to have repeated stimulation. So how do we measure this? You measure this with a myogram. And it's myo means muscle, gram for graph, shows you tension over time. So what are the phases of a twitch? Twitch has a latent period where that action potential is moving across the cell membrane, the sarcolemma, and that sarcomere or sarcoplasmic reticulum is releasing its calcium ions. Okay, then after some point, you get contraction. When those calcium ions bind to that troponin and the cross bridges form. Okay, hopefully that's reinforcing kind of what we talked about before. You get to increase in tension, but it rises to a peak. And in the relaxation phase, the calcium levels decrease. Cross bridges are going to then detach. You get decreased tension. Okay, so what does that look like? So again, tension over time, a different skeletal muscle. So eye muscles have a very fast twitch. Okay, gastrocnemius, a little slower, soleus, much slower. All right, and you'll learn about these muscles, okay, if you're not familiar with them when we get to lab, when we're covering the muscular system. But they all have very similar peak tension, just vary in the length of time that their twitch happens, okay? So that should make sense. I mean, if you think about moving your eyes, you know, you need to be able to move your eyes pretty rapidly, right? You don't necessarily need to be able to move your calf muscles as fast as you move your eyes. All right, so here's kind of a classic graph or myogram tension over time. So here's your resting phase, which happens before the stimulus. And you get a stimulus before any contraction happens, that's the latent period. Once contraction happens, you're in a contraction phase till you get to peak or maximum tension development. And then when the tension decreases, you're in the relaxation phase. Okay, different kinds of twitches. One type of twitch is called TREP. And TREP rhymes with step. Okay, so if you see stepwise increases in tension, it's TREP. Okay, and this happens when you get repeated stimulations right after the relaxation phase. So here, the frequency of the stimulus is going to be less than 50 per second. You get a series of contractions, and you get increased tension. You'll usually see TREP in cardiac muscle, not in skeletal muscle. Okay. All right, for wave summation, you increase the tension because the twitches are being summed. And you cause that by repeated stimulations, okay, before the end of that relaxation phase. Okay, you also have a stimulation frequency of less than 50 a second. Okay. So here's comparing TREP versus wave summation. So TREP says it's an increase in peak tension with successive stimulus. And it's delivered after the completion of the relaxation phase. Okay, that's the key. After the completion of the relaxation phase. So if you get a full relaxation phase, you have TREP. If you have another stimulus before the relaxation phase is completed, that's wave summation. Okay? And you get a larger increase in tension. Okay, so that's a big difference. Now, another term you need to know is tetanus, and that just means maximum tension. All right, so an incomplete tetanus, the muscle's giving you near maximum tension. And this happens when you get rapid cycles of contraction and relaxation. And in complete tetanus, you have a higher stimulation frequency, and that eliminates the relaxation phase completely. You get a continuous state of contraction. All the cross bridges are going to form. All right, so what does that look like? So if you see the graph where you get increasing, so it looks kind of like wave summation up to this point, but when you get to, if you see sustained peak tension, okay, but you have essentially peaks and valleys, that's incomplete tetanus. Okay, and you have brief relaxation periods. Okay, so tension rises to a peak and the periods of relaxation are very brief. But in complete tetanus, your stimulus frequency is so high, your relaxation phase is eliminated. Okay, and tension plateaus. Okay, so like in social studies, you might have heard about the plateaus. So you get a more of a flat line. Okay. Okay. So, all right. So what I'd like for you to do at this point is to explain to your partner the difference between TREP, wave summation, incomplete tetanus, and complete tetanus. All right.